It's my greatest pleasure to be here with you in this both important and exciting week for neglected tropical diseases. Over in Geneva, the World Health Assembly has resumed, and that's where the recently launched Neglected Tropical Diseases Roadmap, taking us to 2030 goals, is being presented to member states for their review and consideration. And amongst many things, the report builds on the incredible successes in reducing the burden of many of these diseases worldwide. And the roadmap also sets out a very ambitious agenda for the next decade. So a really exciting week and delighted to be with you today. Now, at the same time, we're also seeing some incredible and substantial improvements in technology, and in certain fields of science are opening up incredible new avenues for disease prevention, treatment, and control that couldn't be developed or even dreamed of even as recently as a few years ago. So today, in this ISNTD Connect, we're going to delve into Parasite genomics, one of the most exciting fields of science for the future of NTDs. And for this, it's our greatest pleasure to welcome Dr. Gabriel Rinaldi, Senior Scientist in Parasite Genomics in the Berman Group at the Wellcome Sanger Institute here in the UK. So good afternoon, Gabriel.
Good afternoon, everybody. Thank you very much for the nice introduction. It's a pleasure for me to be here.
Our pleasure as well. And you're joining us from Uruguay.
Yeah.
So this is really wonderful. And today, Gabriel, you will be speaking to us about the huge opportunities in this field for NTDs in a presentation looking at the post-genomics era for NTDs. And your background is really interesting. You trained as a doctor, but moved into the fields of molecular parasitology and functional genomics. And from there, you've developed gene silencing approaches, namely for trematodes, including liver flukes, and you've also worked on schistosomes.
Yeah.
And in your current position, you're now supervising a really comprehensive functional genomic platform developed for schistosomes, which usually facilitates the study of parasite genes in their host-parasite interactions. So we are hugely looking forward to hearing all about this, Gabriel. And I'll now hand over to you. And with once again, a very big thanks from all of us at ISNTD. And just before I leave you to it, I just also would like to extend a massively warm welcome and greetings to our many attendees from around the world. Hello to Roland Bamu and Harry de Koning, Majid Harandi, Jafar Ali, and many, many more. Welcome. I can't name everybody, but thank you for joining us, and it's a pleasure to see you today. So with no further ado, Gabriel, I'll hand over to you, over to your presentation. Thank you again.
Thank you very much, Marianne. It's a pleasure for me to be here. First of all, I would like to thank the International Society for Neglected Tropical Diseases, and in particular, Marianne Comparative, who actually approached me and invited me to talk today. It's a pleasure for me also to share with you some of the research we are carrying out in the Matt Berryman's team in the Parasite Genomics group at the Wellcome Sanger Institute in Cambridge. And what have been the main contributions of genomics and post-genomics to neglected tropical diseases, in particular parasitic worms, and in particular within the parasitic worms, flatworms like the blood flukes, schistosomes. Without further introduction, I just want to go through briefly what we are going to be talking about, just to remind you a little bit about what schistosomiasis is. I don't have to remind you what a neglected tropical diseases are. And then I'm going to go into what the meaning of post-genomic era, what does it mean to be part of this era of where we have draft genomes available. And then moving to our latest contribution on functional genomic tool development, ways to manipulate the genome of parasites, in particular, and as an example, schistosomes, including transgenesis and genome editing CRISPR-Cas to induce site-specific mutations. So two kind of cutting-edge technologies I don't know if you guys can see my cursor here, but this is an example of single-cell transcriptomics and what we can learn from them, and induced site-specific deletions in a particular locus, particular gene of Schistosoma mannsoni. So no need to remind you what the neglected tropical diseases are, of course, but I think that it is important to put some numbers in context. Keep in mind that more than 1 million people all over the world are affected by at least one neglected tropical disease across almost 150 countries, if not more. This information was obtained from the WHO website, and this represents not only a huge public health impact in these countries, but also an economic impact, because these diseases are long-term, chronic, debilitating, physical and mental debilitating diseases that, of course, affect the production level of the people affected, feeding into this thing we call a cycle of poverty. So, controlling this disease will eventually also control this, or try to reduce or cut this cycle of poverty. And you can see I color code the different kind of agents inducing these diseases, producing these diseases, like viruses, bacteria, protozoa, a lot of them. But in particular in green, I highlighted the diseases produced by parasitic worms. And just a few examples to show you how debilitating these diseases can be. I highlight the Guinea worm infection, the river blindness diseases by Onchocercas. These people, they get blinded because the larvae induce fibrosis in the eye, and these people can no longer work after getting infected. Filaria with this huge edema caused by this nematode worm. The hookworms, very common not only in Africa but also in Latin American, all the soil transmitting helminths, and in particular schistosomiasis, chronic infectious disease by the blood fluke, schistosomes. I'm actually working on that particular entity. Just to remind you how severe and how spread this disease is. Schistosomiasis is produced by the infection of the 5 main species of schistosomes that infect humans. In particular, 3 of them are the most common. Schistosoma Schistosoma japonicum and mekongii, these 2 small eggs, very common in Southeast Asia. And then we have Schistosoma hematobium and intercalatum, mainly in Africa, Middle East. Schistosoma mannsoni in green in Africa, and also places in South America. More than 250 million people are believed to be infected, but that more than 500, not even 700 million people living in endemic areas, so constantly at risk of getting infected. As I said, the mortality is not so huge, but people can get really sick, up to 20 to 50 million cases of severe schistosomiasis. And not only they die by, the schistosomiasis itself, but also associated pathologies. It's known, well described, that the chronic infection with Schistosoma hematobium is associated with the development of squamous cell carcinoma of the bladder. So that's very interesting from a scientific point of view, but also very worrying relationship between the infection and the development of cancer. It's also important to keep in mind that this infection got spread throughout different parts of the globe. And since 2013, really, there was an outbreak of schistosomiasis in Corsica, a French island in the Mediterranean. And since then, schistosomiasis remained endemic in that area. Nowadays, genomic studies point out to the fact that these parasites are hybrid between hematobium and bovis, but there are ongoing studies on that to actually trace back where these parasites originated in the first place. But the problem is that here I highlighted where Corsica is. In all the south coast of southern Europe, the snail, that is the vector that propagates these infectious diseases, is present. species of Bulinus snails are there in Portugal, Spain, south of France. So there is a potential for this infection to get spread deeper into the continent. And that is a worry, in particular with all the climate changes and so on. And the other important thing to highlight is that there's only a single drug. There's only one drug, praziquantel, that has been used since the 80s and is being currently used in mass administration programs. So there is a really concern of praziquantel resistance, and there's evidence that there's a foci in different regions in Africa where there's low susceptibility to the drug and also resistance. So we are desperate to get new drugs, new control strategies, and eventually vaccines to treat these diseases. And we can do that from many, many different areas. And one of the areas I want to highlight today is the genomics. We can actually contribute with our huge data sets on investigating the genomes of these parasites and find out tentative targets for drug development. Schistosomes entered the post-genomic era, if you want, in 2009, when the draft genomes of Schistosoma japonicum and Mansoni were published in the same issue of Nature. First author of Mansoni genome was Matt Berryman, the PI of my team, and since then he has been constantly curating and improving the genome, not only for Schistosoma mansoni, but also other different blood flukes and different parasites, including nematodes. And they put together this amazing data set in a website called Worm-Based Parasite, where everybody can go into this website. So that's the reason why I leave the website address there. It's an amazing data set with right now 187 genomes representing 161 species. And it was updated last October. So you can find here genomes of all species of nematodes, platyhelminths. You just need to type here in the, you type here in this searching tool the name of the parasite you are looking for, the ID of the gene, and you're going to have immediately information about what you're looking for. You also can download several genomes. You can compare the genomes among each other and build phylogenetic trees and find interesting features of the genome, but also the biology associated with the genome across many, many species. And this is just an example of one project recently published in 2019 about how strong the comparative genomics can be in order to identify a special feature for different parasites. Here, this is a phylogenetic tree, very complete. We have the two main phyla of parasitic worms. Here in this side of the tree, we have all the nematodes, parasite nematodes, all the different clades of nematodes parasites. And here in orangish and yellowish, we have the flatworms. And you can see how well all the trematodes cluster here and the Schistosomatidia family in this cluster. And these are the cestodes or tapeworms. And many, many features are described per cluster and compared among each other. So if you want to read about a comparative genomic study, go to this paper. And kind of in a timeline, I put together when the different genomes were available, starting in last century, if you want. In 98, we got the genome of C. elegans, some model organisms for nematodes and many other systems. In 2007, we have the genome of the planaria, a free-living flatworm that is used in comparative studies with the parasite, its parasite relatives. And then In the 2000, we have an explosion of genomes. Bruglia, the filarial parasite in 2007 was available. Also some plant parasites in 2008. All the schistosomes, the main schistosome species, Mansonella japonicum, as I say, first genomes in 2009, 2012. The hematobium genome was available then, was a better and improved versions later on. Ascaris lumbricoides published a first version in 2018. Other worms, 2013, Amoncus, 2013, tapeworms. And you can see how this map gets completed and complete all over the years. Fasciola hepatica, 2015, the rat lungworm, 2018, and so on. These data sets are being built, getting growing, and they're just waiting for people to get into the data sets and analyze them. This is a free, open, available data sets for everybody interested on these genomes and study biology. Just to give you a small flavor on what we can learn about these comparative genomic studies. For instance, there's a very interesting biology in the Ascaris worms. You know, sometimes when a particular tissue gets into, start to differentiate into, when cells start to differentiate into particular tissues and cell types, usually what happens at the molecular level is that some genes are switch on and some genes are switch off. But the DNA remains the same. So all the cells, for most of the cells, have the same genome. But the difference among the cells is what are the genes that are on or off. Ascaris is a little bit different, in particular for the germline. What these parasites do is just they get rid of chunks of DNA. It's an amazing biology. It's just pieces of chromosomes are lost in the germinal line, and that's the way that cells now know that they need to become germinal cells. That's just an example. And by looking at the genes present in this particular family, there's an expansion of genes involved in chromosome function, in kinetochore function. So there's a really nice association between a known biology and what we find in the genome. And this is actually, each of these bars represent copy numbers of a particular gene family. And you can see the expansion of this family in these Ascaris. Another example of expansion is this family of apirase, proteins involved in immune modulation of the host, really expanded in the hookworm parasites. Then in Strongyloides, we have also expansion of esterase kinase that also are involved in transitions between free living to parasitic stages in this interesting nematode. And you can see an expansion of this family in this clade of nematode, also living in clade 1, is a Trichuras. So you can see how there's a nice correlation between known biology or even unknown biology and something we just described with what happened at the genome level through these comparisons. And then, interestingly, I highlighted this particular family of genes that are expanded in flatworms. These are sulfotransferase, and for some reason they are expanded in trematodes, in flatworms, because these are actually cestode, but in trematodes and in schistosomes are really expanded. Nobody knows what they do, but we know that one member of that particular family of proteins is involved in drug resistance to a particular drug called oxaminiquine. And I like to highlight this because this has been a target for our genome editing approaches. I want to talk a little bit later on. And then, again, from this paper, you can see, you can compare what is known, what is present, and what is absent in the genome of all these worms. And you can study metabolic pathways, for instance. What is present and what is absent in terms of all the different enzymes involving all the different pathways represented in this cell in the right, and you can see in which clades some particular pathway was lost or was expanded, So all these kind of approaches you have access now to investigate and play around. However, these kind of approaches give us a whole picture of what is present and what is known and how that can be linked to the phenotype, if you want. But in order to predict gene function and know what the genes do, we need to go a little bit further and move from the genotype to the phenotype, if you want to know what I mean, a little bit further, and maybe not only study the genome, the DNA, but also the RNA. What are the genes that are expressed in a particular parasite, in a particular developmental stage, and nowadays we can say what are the genes expressed in a particular tissue or cell population. So in order to get closer to the gene function, we need to study the transcriptome. That is, what are all the genes that are expressed at the same time in a particular tissue, and hence being translated into proteins that's going to build a phenotype. So we have now the technology to grab one parasite, ground the parasite, isolate the RNA from the parasite, and sequence that RNA and see what are the genes that are expressed in a high throughput manner. So this is an example of that kind of approaches. I want to share this paper that was was published last May, where we did perform a time course experiment with Schistosoma mansoni. We infect mice with Cercaria, infectious larvae of Schistosoma mansoni. At the different time points after infection, we collect the parasite. At day 6 after infection, it's known that the parasite is migrating through the lung. It's a pretty unknown stage, and nobody really knows what's going to happen, what is going on within the lung for this blood flux. After the lung, the parasite starts circulating throughout the body within the blood vessels. It's a blood fluke, and at day 21, they reach the liver. So at different time points, we collect the parasites from the blood from day 6 to day 35. And then we study the parasite, how do they like, if they are big, small, we categorize that, and then we study transcriptome. I say we, but most of this work was done by the first author of this paper, Arpon. Coy is her nickname. He got his PhD a few years ago out of this work. And first thing she did was, after collecting the parasite from different stages, she classified the parasite based on this staging schematic published in 1934. And you can see in this paper, they isolated worms at different time points, and they classified the parasites, depending on the size, on the development, in 21 discrete categories. So Coy actually collected the worm from all these different time points, and she classified them based on exactly the same staging system. So each column represents percentage of parasites within each category, and each row are the pool of parasites collected from individual mice. For instance, in day 6, in this particular mouse, we have that almost probably 80% of the parasites belong to the stage 4. And you can see in the lung stage, the vast majority of the parasites belong to stage 3 and 4, are these tiny worms here. But then, and this is very interesting, and this is all biology, all known biology, after the parasites, they leave the lungs, they start to get asynchronized. Some of the parasites develop faster than others, and some get a little bit stuck. And you can see by day 13, 17, and 21, when the parasites circulate, they get all scattered in terms of size until they reach the lung, they reach the liver at this point, and they start to pair up, male and female, and produce eggs. So this is a really nice piece of biology she put together, confirming previous findings. But what she did on top of that, she isolated RNA from each of these stages, and she studied the transcriptome. And one way to compare transcriptomes among each other is by doing these kind of plots, where you can see all your data, and these are principal component plots, where each dot is the transcriptome signature, let's say the transcriptome of a particular pool of worms collected from a single mouse. So the only thing we need to know at this point is that the closer the points, the more similar the transcriptomes are. The closer the points are, the more similar the signature, or the same genes are being expressed, the same amount, etc. And this is really nice because you can see how all the worms collected from the lung cluster together very nicely, as expected. But then you have the worms collected day 13, day 17, 21, that kind of cluster together following kind of a trajectory that actually parallels the biology. And then we have clustering far away from these two, day 28 and 35, when the worms actually pair up. And we know that pairing, male and female, trigger a lot of transcriptome changes in both sexes. And day 35, these are transcriptomes full of genes related to egg production. But in particular, we want to focus on the lung stage. That is kind of really unknown what's going on there. We know that these blood flux go through the capillary beds of the lung, and you can see in this patient that inflammation around a migrating worm. And so she's compared what's going on in this stage with the other stage, and you can do these kind of comparisons, asking what are the genes that are upregulated in this stage compared to this one, and vice versa. And she came up with this really nice table with the top 15 or top 20 genes that are upregulated or overexpressed in the lung stage. So we can know what's going on in the parasite side of the equation in terms of what are the genes that are expressed. And many of these genes are secreted proteins. These particular genes that are called microexon genes, particular genes of Schistosoma mannsoni that are excreted in the lung. We have no idea so far what they actually do. So this opened a huge set of questions to follow up. And there are beautiful analyses he did in the experiment. I am kind of biased, so that's the reason why I say beautiful, but you might find this boring. But I kind of get excited about this kind of analysis because he managed to cluster the different genes based on the expression profile. So in these little squares, we have in the x-axis, we have the time points from early worms, lungworms, to late worms, mature worms. And then here in the y-axis, we have expression levels. So you can see that many genes cluster together based on how similar the profiles are. For instance, in this cluster, you have genes that are highly expressed in the lung, but then the expression drop later on and remain really low for the rest of the cycle. Some of the genes show a high expression during the migration, circulation, but not in the lung, not in the other worms. And other genes like this one down here, they have a low expression in the lung and increased expression. So we can go, if you want, one by one, see what are those genes and see what is known about those genes. But these are what we call bulk transcriptomic approaches, where we ground a whole worm full of cells, full of tissues. Remember, these are multicellular organisms. And the expression level we can measure at the end is an average of all the genes produced by all the cells, by all the tissues. So there must be many, many genes with low expression or genes expressed only few cells that are lost in this kind of analysis, cannot pick up. So that is when this from fruit smoothie to fruit salad metaphor came into play. And this is, I think, a really nice way to understand the genomics and the expression studies in a nice metaphor. Let's say we have a complex tissue full of different kind of cells and different kind of tissues, in this case represented by different kind of fruits. So we have grapes, we have bananas, we have apples in different amounts. When we do a bulk RNA-seq study, as the one I just showed you, what we do is we mix all these fruits together in a blender, we blend it and we make a smoothie. So bulk genomics and bulk transcriptomic studies is like drinking a smoothie. You can drink it and say, yes, it might have a little bit of strawberry, it might have a little bit of kiwi. I don't know how much kiwi or how much strawberry. So it gives you a big picture of what is expressed. Then we have an intermediate approach where you can actually, before doing the sequencing, you can actually sort out the different tissues. Let's say instead of grounding a whole worm, you can separate male and females, and then you can cut the head of the female, cut the ovary, and just sequence the head, sequence the ovary, so you just sequence specifically particular tissues. And this is what is represented here. You can do cell sorting, let's say enrich for cells of the neurosystem, and sequence just that population. So you are getting closer of what are the fruits present in the tissue in this case. But lately, and this is in the last 5 years probably, is where this single cell omics, and in particular single cell transcriptomic technology, exploded. And that was a fantastic opportunity for us to explore this platform in worms. In this technology, you actually separate the worm or disassociate the worm into individual cells. So you have a parasite with hundreds of thousands of cells separated, so you treat the parasite with different kind of enzymes or mechanically disassociation. That is a long optimization protocol. And then you, after you get the individual cells, you use these technologies to sequence the transcriptome of individual cells. So what are the genes that are being expressed in one cell? And then using different kind of algorithms and software, you can actually cluster the different kind of cells. So you will be able, in this case, to know how many strawberries you get. Not only that you have a strawberry, but how many you have in the pot, or how many bananas, or how many pieces of kiwi, etc. So it's really, really sensitive and specific. And then the main question is, once you have individual cells, is where the cells are expressed in the whole tissue. So you need to use some kind of approaches to label these cells in the whole tissue. And this is exactly what we try to do in this project, where that produced a paper that has been recently accepted in Nature Community of the schistosome in the mammalian stage. It's called schistosomula or juvenile schistosomula. We disassociate the schistosomula into individual cells, select by fax only the live cells by sorting, and use a technology that allows to actually introduce one single cell per drop of oil drops, and all the transcriptome reaction happens in a single drop. So you have now all the information of all the genes that are present in that particular cell, and then we identify these cells in the whole organism. So these are the kind of maps you get from single cell seq. I'm not going to get into details, but we managed to identify more than 13 cell clusters in a sommule that is a very simple organism with 1,000 cells per worm. But within that worm, we managed to identify 13 individual clusters and subclusters. For instance, the tegument, we found 3 subtegumental clusters, a big cluster of germ cells, 3 different clusters of muscle cells. So many cells already committed to differentiate in different tissues in this early stage. These are 2 days old sommules. And to do that, we identify different markers for each cluster. So each cluster has the top 1 or top 2 genes highly expressed in that particular cluster, and those markers define that cluster. And we use a technology called in situ hybridization to map those clusters, those markers, into the tissue. And just to give you a flavor, this is the way we validated some of the clusters. So we identified a muscle cluster that we call positional muscle, because the main marker was this gene called WIN2. And you can see in the sommules, using this in situ hybridization technology, we found that the gene is expressed in the head of the somule, but later on in the development, there's a gradient of expression from anterior to posterior side of the worm. So that's the reason why we call positional muscle. So it doesn't have anything with the muscle itself, but set up the anterior posterior axis of the worm, or at least this is what we believe. Then we found a specific cluster for MEG genes, those genes that I showed you before that were upregulated in the lung stage. There's a specific cluster that is a subtegumental cluster, so the same origin that the tegument, that is a part of the esophageal gland and is rich with macroexon genes. So somehow this parasite probably is getting into the lung and expressing from the esophagus a lot of proteins encoded by these microexon genes. Nervous system, we found 3 clusters of nervous system, and you can see how this particular gene that is expressed in this tiny region in the somule, in the cephalic ganglia, is expanded in the worm. So all these cells that are confined in this region in the somules get expanded in the rest of the nervous system of the adult worm. And the stem cells is a big issue as well. We identify a stem cell marker. The earliest stem cell marker detected, that is a calmodulin, and we are currently investigating what this marker is doing, maybe defining sex, male and female phenotypes, who knows. But this gene that is expressed in 5 cells, only in 5 cells in the somules, are then later on in the development get expressed either in the testes or in the ovary of the adult worms. And right now we have an ongoing project of doing single-cell-seq across different stages, including, so meaning we are trying to build a cell atlas across the stages, and we have a collaborator in the States, Jim Collins, who has recently published a cell atlas for the adult worms, and that was published in Science a few weeks ago, and he describes single clustering for male and female worms. And so we, in collaboration with him, we are trying to get cell atlas for all the stages, and this is just a preliminary data from single cell from sporocyst. So we can culture intrasnail stages in the lab, disassociate them, and get some nice single cell clustering. But in order to actually define the role of a gene, of a protein, we need something more. With the genome and with the transcriptome data, we can have an idea, or we can generate hypotheses of the function of the genes. But in order to validate the function or proof or disprove a hypothesis of what is the function of a particular gene, we need to be able to manipulate the transcriptome and eventually the genome. So we need to knock down genes or knock up genes. And so far, and after so many years, the only tool that is being employed massively, routinely in many labs is RNAi. RNA interference, that, as you may know, is a tool to knock down genes at the messenger level, to get rid of the expression of a particular gene and see what happens with the phenotype, is still a tool that is being used since 2003, where these 2 papers came up more or less at the same time, showing that RNAi is active in schisto. And since then, hundreds of papers on RNAi has been published. The most recent one, again, from our collaborator, Jim Collins, in the same issue of Science, he published for the first time this large-scale RNAi screening against Schistosoma mansoni. He tried to knock down almost 3,000 genes, and he described here the protocol he followed to knock down genes, but also some of the phenotypes he described for the knockdown parasites. So this is a huge, groundbreaking study where we show for in the first time that large-scale screening can be done. Having said that, and knowing this, how amazing this study is, he found that only 10% of the genes show a phenotype. Out of 2,000 and something genes, he found phenotype only in 200. no, it's not only, it's not a bad thing, it's just a data from the reality, meaning that 90% of the genes he tried to knock down, he didn't see any phenotype, meaning that the gene couldn't be knocked down. We know that there are many genes that are refractory to RNAi, or the gene was knocked down but didn't produce any evident phenotype. So in addition to RNAi, we need to move forward and try to apply novel technologies that could be employed to knock down genes that cannot be knocked down by other means. In the past, I've been trying in Paul Brindley's lab in Washington, D.C. to develop transgenesis, and we use retroviruses to transfect the parasite, and we managed to identify and to prove that transgenes get integrated in the genome of the parasite. These are integrations of the transgenes across all the chromosomes of Schistosoma mannsoni, and that transgenes got propagated throughout different generations, meaning that we managed to produce transgenic lines. The big problem at this point is that all the transgenic lines we produce, and was more or less 5 or 6, they got silenced. So retrovirus is a good way to introduce transgenes, but they get silenced. And this was published in 2012. And since then, we couldn't overcome the problem with the silencing. We also found that lentivirus can transfect schistosomes. So even though it seems to be promising, there are a lot of things to work and to optimize before applying this technology routinely. And as you know, the latest news on genome editing, and this has been also stimulated by the Nobel Prize, given that one of the two main researchers involved in genome editing via CRISPR-Cas received the Nobel Prize this year because of this tool. And we have been exploring using genome editing in schistosomes, as only a few parasites have been knocked out by CRISPR, like Strongyloides, and that's it. And we are trying to do schistosoma as far as I know. So briefly, for those that are not familiar with this technology, I'm not going to get into details, but basically, this CRISPR-Cas technology allows to induce site-specific mutation. So you know particular place of the genome you want to mutate, and you just introduce a mutation. How do we do that? So we use a nuclease, DNA nuclease, called Cas9, that is linked to a guide RNA, and this guide RNA encodes the sequence of the region you want to target. So basically what you need the sequence of the locus you want to target, 20 nucleotides, and you need an enzyme. And then you transfect your parasite with this enzyme, this ribonucleoprotein complex, and then if everything goes fine, the enzyme will find this place in the genome, will induce a double strand break, and then the DNA repair mechanism of the cell will either quickly combine the ends of the breaks, producing what is called a non-homologous end-joining repair, NAGJ, that usually introduce indels, insertion and deletions in that region, but that's enough to induce a mutation. Or if you provide a template DNA with homology arms, you can drive a knock-in via homology direct repair mechanism. So you can, in addition to the ribonucleoprotein complex, you introduce a template, and you can actually knock things in to the genome in a site-specific manner. So the first thing we did, we studied if the pathway, the CRISPR-Cas pathway, is present in Gisto, because we have the genomes. And effectively, we found that NAGJ pathways and HDR pathways are present in Schistosoma mannsoni. Interestingly, the expression profile of these pathways is different depending on the pathway. This is RNA-seq from different stages, from eggs to adult worms, of different proteins in the NAGJ pathways. And you can see that in general, there's an the older, the more mature the worm, the higher the expression of this pathway. Probably because the worm is the worm faces a lot of insults from the host and the environment, from the immune system, and they need to keep repairing the DNA. Interestingly, for the HDR, there's a huge peak of expression of this pathway in the intrasnail stages. And this is probably because, as you might know, in the intrasnail stages of this parasite, there's a lot of replication. So these pathways that is overexpressing in dividing cells need to be active. So what we did is we selected a target, and the target we selected is this sulfotransferase I mentioned at the beginning of the talk, because we know, and has been described in this paper, that the sulfotransferase is an enzyme that is very important to activate this prodrug into an active drug. This drug is called oxaminiquine. So when the prodrug enters the cell, the sulfotransferase sulfonate the molecule, activate the drug, and this will kill the parasite. And we quickly try to reproduce some of these experiments. We knock down by RNAi the sulfotransferase. So we have 3 groups of worms, and one group of worms we didn't treat with anything. Another we treat with an irrelevant RNAi molecule, shouldn't have any effect. And then another group of worms we treated with double-stranded RNA against sulfotransferase. So we did an RNAi experiment and knocked down the protein. And then when we compared what happened with the worms in the presence of the drug, in the mock, all the worms were dead. In the irrelevant control, some of them were still alive. So we partially reproduced the findings in the paper. But in those worms for which we have a knockdown of the enzyme, you can see they don't care if there's oxaminiquine because they became resistant. So we wanted to use this locus as a proof of principle that the CRISPR-Cas technology works. I'm not going to get into details on how we designed the experiment, but basically this is a region. So the sulfotransferase has 2 exons, coding regions, and we designed our guide RNA targeting the first exon at the beginning of the exon. And then we bought from a company Cas9 protein and the guide RNA that was conjugated with a fluorochrome. So we can actually treat the worm and see in the microscope where the molecule went. And we deliver the molecule by electroporation. So we electroporate eggs, sporocysts, and worms, 3 different stages. And just to let you know, this has been published a few months ago in Wellcome Open Research. You can go and read it, but basically you can see how the molecule, even though got stuck, a lot of molecule outside the worm, some of the molecules got into the cells, and we can capture those cells here, also in sporocysts, and few in the eggs. And then we isolate the DNA from these worms, and by PCR, so we amplify this region and we sequence the region and looking for mutations. Interestingly, we didn't find any insertion, we didn't find any substitution, we did find deletions. So somehow, by treating these worms with this ribonucleoprotein complex, CRISPR-Cas, we induced deletions, and some of them big deletions. I'm not going to get into details here, but this is just a representation of all the sequences we produce from these treated parasites. And this is the first line, is the sequence of the wild type allele, wild type gene, if you want. And all the rest are the sequence of the mutant alleles. We managed to introduce all these deletions in this region of the genome. And this is another way to look at this, where we have in the x-axis the region where we target in the sulfotransferase gene. This blue line represents the double-strand breaking predicted site, and these horizontal lines represent different all the deletions we found, and color code represents the number of reads, number of sequence reads that support each of these deletions. And these are controls. You can see clearly how many more deletions were found in worms or untreated parasites compared with controls first, and many more deletions were found in worms compared to sporocysts, and then almost no deletions at all in the eggs. So this is an ongoing work. This was a somatic experiment, so we expect only somatic cells got mutated, and hence we didn't see any resistance at this point. We didn't even see a knockdown at the messenger level. But this is published. It was the first description of CRISPR-Cas across all these stages in Schistosoma mannsoni. It was a first paper published in eLife last year, where, and we were collaborators, they found CRISPR-Cas in eggs inducing a particular mutation. In here we try to apply the technology across other stages. So, to conclude, just to remind you and put the numbers in perspective compared to what happened with coronavirus, we have more than 1,000 million people infected with at least one species of worms. We have this problem of drug-resistant development, so we need to, we are desperate to develop new drugs, and genomics and functional genomics would be strong fields to contribute toward this aim. Basic knowledge of helming biology that we learn from all these studies, at some point, needs to be translated into control strategies. So from all these targets we found and are interesting based on the biology, we need to find a way to transform or to translate into control strategies. For instance, defining new targets for new drugs. Some of the genes expressed in the lung, maybe they have drugs or inhibitors that we can be exploring in the future as a new new targets for drugs. As I say, single-cell transcriptomics allows the identification of key genes, so it's a cutting-edge technology that will help to learn a lot about all these parasites. The same with the genome manipulation. Even though we know it's feasible and CRISPR-Cas9 seems to be active, we still use RNAi as the main tool to address gene function. And with that, I just want to thank all the people involved in several of these projects, my lab mates, workmates in the Parasite Genomics team, starting with Matt Berryman, of course, but all the people who actually ran the hard work in the lab, also the animal supporting team at the Wellcome Institute, and our collaborators overseas, and of course, the funding sources of our work. And with that, I'm done. Happy to address any question.
Thank you so much, Gabriel. I think quite a few of the attendees are joining themselves to me and giving you a very big thank you. Franklin Aisi from the University of Ghana saying so educative and insightful talk. Jackson Wema as well, nice talk, Gabriel. Deborah Adipoju, great work, hope to replicate. So certainly that's been really amazing. Thank you so much, Gabriel. That a huge amount of work there, massive body from an incredibly respected institution as well, the Wellcome Institute, which will really help us to understand the genes behind some of the major traits of parasites, which can hopefully be then exploited for new interventions. So thank you for that. I should also mention that probably like a lot of our attendees, I do have a smoothie often with my breakfast, and so I will never look at my smoothie in quite the same way. I'll always be thinking of bulk genomics. So thank you for that.
That's good.
So we're almost actually there was so much there to explore and delve into that we've nearly reached the end of our webinar. However, there are a few questions that have trickled through. If you don't mind, Gabriel, if you do have a few minutes for us, well, then that would be brilliant. Thank you so much. And maybe first and foremost, in no particular order, but we had here a question from Fiona Allen, which perhaps you know as a fellow Londoner from the Natural History Museum here in London. And Fiona's working on schistosomiasis as part of the parasites group there at NHM, and was wondering, do you think that the definitive host used in some of your work might impact the development, i.e. mouse model versus human or other larger mammal? And this was also a question slightly echoed here by Deborah Adepoju, who's asking the gene expression in mouse experiments, was it compared to the same infection of schisto in humans? And actually,
that's a brilliant question. And
before you answer, Gabriel, sorry, would you mind to actually, I can probably do it from here if I just take off your screen share. There we go.
Yes,
now
yes,
we can see you.
Yeah, that's a really good question. We ask ourselves that question when writing the first paper, and we were asked by the reviewers, and the answer is probably yes, or yes. The host you use to do the experiment definitely will affect the expression profile of the parasite, and there are few papers, previous papers, where using a different platform, like I think was microarray, and I think one of the authors was Chef Gobert, that is currently in the Queen's University in Belfast. I think he's one of the authors, or I might be wrong, but he did microarray experiments in worms collected from mice versus worms collected from rats, and in Japonicum, and it's known that for some reason rats are more resistant to schistosoma infection. So they were interested in this kind of phenotype of resistance of the host against schistosoma, and they did find quite a few differences. So I would definitely expect differences. The problem is that the mouse model is a great model to maintain the life cycle and also to answer specific questions. But in order to translate the findings into humans, we have to be more cautious. The problem is that we don't have access really to intramammalian stages of worm from humans for obvious reasons, unless you get samples from autopsies. But yes, to make the answer short, yes, I expect differences, in particular for those genes involved in the host-parasite interaction.
Thank you very much. And a question here from Pete Olsen, also from the Natural History Museum in London, Parasites and Vector.
I know him.
You know each other, of course.
Yeah, I know them.
Yeah, we don't need too many introductions in this group, I'm sure. But so Pete was first of all saying, great CRISPR results, and what explains getting any deletions at that specific locus in your control worms?
What explains getting any, you mean some deletions? We found some deletions or non-deletions at all?
Any deletions? Why do you think that any deletions were found?
In the controls? Well, there are few deletions in the control, but they are not really present in the double strand breaking site. And the good thing about this study is that we have replicates, and we replicate the experiment at least 2 or 3 times. So 2 for sporosis because 1 failed, but 3 for adults, 3 for eggs, and even for adults we have technical replicates as well. By replicate, I mean we repeat the whole experiment with a new batch of parasites. And by doing that replicate, we were able to do statistics, and definitely there are many more deletions in the treated groups compared with the control. If I can I share quickly one of the slides?
Of course.
To actually let me see. So for instance, these are the deletions we found in the controls. So you can see they are very scattered deletions here and here. Here, these are artifacts due to the priming of the amplicon sequence. There are a few more, in the egg control there are kind of few, but also we have so few in the treated that even though this is significant, I would say that the deletions we managed to introduce in the eggs were so rare that I'm not saying that I don't trust in this data, but I would really look at this data very carefully. However, if you look at what happened in worms and spores, by just looking at these, and these are the deletions found in a single experiment. And here in the tag you have what the experiment was. If you combine them all and combine all the things, deletions found in the control, definitely there are a significant number of deletions in the treated. I don't know if that was the question, Pete. Why we found deletions in the control, but those deletions were really probably not related to CRISPR, and so little.
And Pete's just saying the results are very clear. He was not challenging them, and also definitely agreeing that it just seems unlikely you would see any deletions at that specific site by chance.
Well, yeah, the background deletions we found in the controls. That's what we found. Yeah, I think it's important for just a brief comment. It's very important from now on in all the CRISPR papers to include controls and number of replicates, because sometimes in the very beginning the control we used to use is just the sequence from gene bank or worm-based parasite, and no, the control has to be, as we know, worms treated in parallel in the experiment, treated with no molecule or treated with just Cas9, but we need a control in the experiment itself.
Brilliant. And so we've heard a lot about this, I can say, beautiful science, and a couple of our attendees were wondering about bridging this technology with control programs in the field. So first question here from Professor Majid Fasihi Harandi from the Kaman University in Iran. And Professor Harandi was asking, practically speaking on the ground, how can we bridge the RNAi and CRISPR-Cas9 technology to the control programs in the endemic areas? And in your opinion, Gabriel, how long would that take?
That's also a good question, very difficult to answer. We all agree, I think, that these technologies are really important to move the field forward, because we have been generating these huge data sets with a lot of information in genomic and transcriptomic. We basically need people to mine the data, identify potential candidates to develop drugs. I can give a particular example with Tricuris. There's a project in our team, Tricura, where from that comparative genomic data, the bioinformatician leading this project found tentative candidates on this particular clade of parasites, and she managed to repurpose some drugs against these targets. And right now, we are in the process of testing those drugs in vitro. We have already data, and about to move into in vivo treatment in mice. So we are moving towards, that's an example of how from a comparative genomic study, you reach the in vivo drug efficacy testing at this point. And these are drugs already used in human for other purposes. So it's repurposing drugs already approved by FDA into anti-helminths. But the other question about CRISPR or RNAi, this will allow us to study the function of genes. Basically, it's a functional genomic tool, and validate the importance of a particular gene. I mean, James Collin, in his RNAi paper, he actually described targets for potential drugs. That paper is a really nice example of how, from an RNAi screening, he detected until now unknown targets to develop new drugs. And they are about, in collaboration with Carl Hoffman in Wales, who has a nice phenotyping platform, they're about to screen novel drugs, that all of them came up from this large RNAi experiment. So it looks far away, but there are links, and there are links that started to be built between the basic science and more translational science.
Building on from that, we've had a question here coming in. It's actually from my colleague Cameron Rafiq here at ISNTD, and who was just commenting and saying most people might tend to tie the genomic approach with the discovery and development of novel therapeutics. However, do you think this also has the potential, and what would be the impact of vector control? And is that something that's been considered?
Yeah, I'm aware that there are a few labs, at least 2, trying to develop CRISPR-Cas-based transgenic lines of snails. I don't know what are the latest news on that. I was told at some point there was a Cas9 expressing a snail line, but I've never seen the data. But there's a recent paper from Paul Brindley's lab. They managed to do CRISPR-Cas in VGE cells. It's not a snail, but are the snail cells. And they managed to actually prove CRISPR, they delete a particular locus in the cells. So in the same manner that CRISPR-Cas was used for mosquitoes, I'm pretty sure sooner or later that will happen for snails. From that, the snail genome, even though published, is still pretty fragmented. So you need actually a good reference genome to design your experiment nicely for CRISPR, for instance. And as far as I know, the snail genome, in addition, that is very complex and big, is pretty fragmented.
Mm-hmm. So watch this space then.
Yeah. But I know that there at least a couple of, we thought about that at some point, but we wanted to focus on the parasites at this point. But I'm aware that at least 2 labs trying to develop CRISPR in snails.
Okay, thank you. Conscious we've kept a lot of your time, Gabriel, so perhaps one last question to conclude and round off the discussions today. We have here Maria João Gouveia from the Instituto Nacional de Saúde in Porto. And Maria was asking, in your opinion, what should be the next steps in schistosomiasis research? What are the points that should be focused on? Treatment, elaboration of control programs, and thank you with a smiley face. So very broad,
I know her, and say hi to Maria. I know her.
Of course, you know each other.
Yeah, we work in collaboration in another really exciting field, that is schistosoma hematobium and bladder cancer. Well, I guess that it's it has a kind of a personal side, this question, because I could say that whatever you are interested in, if you're a more like molecular person and you know your skills are there, focus on that. Trying to, in the case of bladder cancer and hematobium, I would say, okay, trying to dissect the pathways present in the parasite, but also in the host tissue that can drive carcinogenesis, for example. I think that each person has to focus on on what they like to do and what they're best to be doing. But it's very important to keep a network and know what the other people are doing, because we cannot be just going to meetings to talk about genomics and only genomic people will be there, because they have no idea what could be the impact of their own research into the field, and vice versa. We have to show how the genomics side of the coin can help to improve the conditions in the quickest probably impact in controlling many of the disease. Because I love genomics, I love molecular biology, but I also understand as a physician in my whole life that it's very, very important to actually address critical issues like social conditions, education, and sanitation. And that's probably one of the biggest challenges we have nowadays. Changing behaviors is one of the most difficult things when you want to approach a condition. I mean, using mask now in the pandemic is difficult. Not everybody like to use a mask, and with those simple things, you can make big changes.
Fantastic, and that's a lovely message to finish this webinar on. That was started off from a very technical perspective, and I think you've married that really nicely with the broader policy and collaborative agenda that really needs to happen around schistosomiasis. So thank you so much for that. Dr. Vanna Haluskova as well in the chat. I can't summarize all your posts, but is also very much talking about online collaboration or having a platform to look at all these things that you mentioned, even including this in climate forums and economic forums, also looking at the issue of resistance. So thank you, Dr. Haluskova, for that as well. On this note, I'd like to thank you once again from all of us here at ISNTD. Thank you for your time, and to all our attendees as well. And please, we look forward to seeing you again very soon, whether as a speaker, doctor, or whether as an attendee, or maybe even one day in person here in London, who knows? Yeah. That day may finally come. In the meantime, all the best, Gabriel, to you, and all the best to our attendees. And keep safe.
Thank you very much. Have a good day.
Thank you so much. Bye-bye, everyone.
Bye-bye.